Well, hello everybody, and welcome to this presentation of the 2022 Q1 results and report for Lytix Biopharma. My name is Ole Peter Nordby. I am the head of investor relations at Lytix Biopharma, and it's my pleasure to introduce our CEO, Øystein Rekdal, who will lead us through this presentation. Please note that the presentation will be available at our webpage afterwards, and that we will take a Q&A session after the presentation. And you can send us your questions during the presentation by clicking at the Q&A button in the upper right part of your screen.
Good afternoon, everyone, and welcome to this first quarter report for 2022. So I'm already introduced by Ole Peter, but with me today we have our Chief Development Officer, Graham Curry, and our CFO, Jess Breistein. So Lytix Biopharma is a cancer drug development company and owns a unique technology platform consisting of a pipeline of promising oncolytic molecules. And the company was listed at Euronext Growth in June last year. Our lead drug candidate is currently tested in a phase 2 study in the US, and in April, a second phase 2 study was initiated in US by our commercial partner, Verrica Pharmaceutical, for skin cancer disease. And before we dive into the first quarter highlights, I would give a brief introduction to our technology and how it is addressing one of the major challenges in cancer treatment. Cancer is still one of the largest health problems globally, and one reason for that is the fact that new mutations occur during cancer progression, leading to a number of different cancer cells within each tumor. This is called tumor heterogeneity. In addition, many of the cancer mutations are unique, making no tumors similar. This diversity of mutation results in different cancer cells within a tumor, different set of cancer cells in different tumors in the same patients, and different set of cancer cells in different cancer patients. So it's a really big challenge to eliminate all the different cancer cells in one patient, and it is almost impossible to develop drugs that work in a number of patients. And among several different challenges, oncologists rank tumor heterogeneity as the major challenge in current cancer therapy. And as long as we are not able to kill all the different cancer cells in a tumor, the cancer will recur, and often with more resistant cancer cells that are even more difficult to treat. So, how can oncolytic molecules be used to solve tumor heterogeneity? First of all, oncolytic molecules are able to kill all the different cancer cells in a tumor, including cancer cells that are resistant to chemotherapy. Second, oncolytic molecules kill cancer in a unique way, resulting in release of all their contents, including the mutations which are exposed to the immune systems. This results in a broad T-cell response that are able to attack the different cancer cells with a unique set of mutations. So, when an oncolytic molecule is injected directly into the tumor, all the different cancer cells will be killed, and the released mutations or tumor antigens will be exposed and transported to the lymph nodes, where all the different T cells are located. And the T cells that recognize one of the mutations or tumor antigens will be activated and multiplied. The activated T cells then enter into the bloodstream, infiltrate tumors in the body, and attack and kill cancer cells which have the same mutation or tumor antigen it has reacted to in the lymph node. So, by killing all the cancer cells locally in a tumor, a broad repertoire of T cells can be activated that can target cancer cells elsewhere in the body, and thus help fighting the entire cancer disease. This is a type of vaccination called in situ vaccination, which means vaccination on site. So let's then dive into the fourth quarter highlights. A great milestone for Lytix in this quarter was the initiation of Verrica's phase 2 study with LTX-315 for basal cell carcinoma, a skin cancer disease. And this milestone triggered a milestone payment of $1 million to Lytix. This milestone also proves the quality of our partner strategy, and with this second phase 2 study in the US, the chances for our leading drug, LTX-315, to succeed increases. With regards to our own phase 2 study, ATLAS-IT-05, that is open in US already. We are currently expanding now to sites in Europe with expertise in intratumoral therapy, with the aim to recruit 20 patients within 2022. In addition, our small phase 2 study in Europe, ATLAS-IT-04, is completed, and results from this study will be presented at ASCO in June, which is the world's largest clinical cancer conference. So this means that LTX-315 is tested in 3 separate and independent phase 2 studies. And our second generation molecule, oncolytic molecule, LTX-401, with promising effects in liver cancer, will be made ready for clinical studies in liver cancer patients within 2022. Our promising approach to solve tumor heterogeneity are being recognized internationally, and in March we were invited to give a plenary talk at the Next Generation Immunotherapy Conference in London, focusing on how oncolytic molecules are addressing tumor heterogeneity. Then we move on to our clinical and operational update. Verrica Pharmaceuticals has in-licensed LTX-315 for certain types of skin cancer, and is focusing initially on basal cell carcinoma as the lead indication for development. And as earlier mentioned, they have already started treatment in patients. Basal cell carcinoma is the most common malignancy in humans, and it is estimated 3 to 4 million diagnoses annually in the US alone. And Verrica's plan is to enroll 66 patients with basal cell carcinoma in their phase 2 study, with an estimated completion of enrollment February 28 next year. I will then let our Chief Development Officer, Graham Curry, give you a clinical update on our own internal clinical program. So please, Graham.
Thank you, Øystein. Just confirming you can hear me?
Yes.
Yeah. So as Øystein mentioned, ATLAS-IT-05 is being conducted at clinical sites in the US. In this quarter, we did amend the protocol, which increased our focus on focusing on patients with advanced melanoma. The goal of this is to ensure that in this small focused phase 2 study, we show signs of clinical efficacy. 2 sites have been initiated, and enrollment is ongoing, and we expect to complete that within 2022. In addition to the sites that we have in the US, we have engaged a larger network of clinical sites through a company called Oncobay, who will participate when they have a patient identified. And the advantage of this approach is a very short site activation time. And as Øystein also mentioned, we have started the process to open a group of highly experienced investigative sites in Europe. The reason why we're thinking about moving to Europe is twofold. One, we've seen that US sites have been impacted by the COVID-19 epidemic, one in terms of patient flow, and secondly, in terms of staffing. We quickly added OncoBay in February of 2022, and we have also reached out beyond the US to highly focused melanoma sites in Europe. Now, while this may take some time to move through the regulatory process, the process now is very streamlined, and we hope that that review will occur very expeditiously, and that those sites will be open later this year. In addition, we've expanded our staffing reach, and we have a new head of clinical operations, a highly experienced oncology-focused individual named Jackie Irbina. Lytix, Øystein mentioned earlier that Lytix has been in a study at Herlev Hospital in Denmark, and it was studied in combination with adoptive T-cell therapy in a very difficult to treat immunologically cold patient population, sarcoma. Adoptive T-cell therapy is a method to isolate and expand patients' existing own T-cells and then make them provide them back to the patient. That means that there is added number of cells to attack the tumor. Soft tissue sarcoma are often noted as very immunologically cold tumors, and that means they have a very low number of T cells. So this T cell expansion exercise is a way of increasing the immune response, and the addition of 315 not only increases the number of T cells in treated tumors, but also has the added effect of ensuring that they're focused against the released tumor antigens. The key objectives of this particular study is to really understand, does Lytix 315 induce T cells that are able to infiltrate these immunologically cold sarcoma patients. We also want to know that whether the T cells are generated specific for those tumor antigens that have been released from the patient's tumor cells. And in this small study, we also wanted to understand whether there's any correlation between T cells generated by Lytix 315 and the clinical outcome. The trial has completed recruitment. There were 6 patients that were completed. Data analysis is ongoing, and we have had an abstract accepted at this year's ASCO conference in June in Chicago, and the results will be presented in a poster presentation at the meeting.
Clinical update. As mentioned, we also have a second-generation oncolytic molecule, LTX-401, which have demonstrated very promising efficacy in liver cancer. Liver cancer include hepatocellular carcinoma and liver metastasis, which both represent 2 large cancer segments with high unmet need. The hepatocellular carcinoma market alone is expected to increase to more than $5 billion in 2029. LTX-401 is in preclinical development and seems to be well tolerated, and maximum tolerated dose has been identified. The preclinical program for LTX-401 is expected to be completed within 2022, and preparation for the phase 1 study in liver cancer patients is ongoing. This development project has received 3 million NOK from a regional research fund, RRF. If we look forward on the key objectives for 2022, we are, as Graham mentioned, expanding to European sites with aim to complete enrollment in the Atlas IT-05 phase 2 trial by the end of 2022. We will continue to provide Verrica with LTX-315 in API form, which mean the active pharmaceutical ingredients, and support them in their phase 2 trial in basal cell carcinoma. As mentioned, data from Atlas IT-04 will be presented at ASCO in June. We look very much forward to that. And LTX-401 will be made ready for applying for a phase 1 study in Europe. We will continue to be present at international investor conferences and to increase attention within the investment community. And we are looking for additional opportunities to expand our innovative pipeline of oncolytic molecules. Then I will let our Chief Financial Officer, Jesper Eistein, give a financial update for the first quarter of 2022.
Thank you, Øystein. First quarter of 2022 has been a very good quarter. We have had a lot of activities ongoing. We've mentioned them all here today. We have good progress on the Verrica collaboration. We are on good way with Atlas IT-05 and the Atlas IT-04 trial as well. And not to mention the LTX-401 program, bringing that molecule to the clinic. And we can see this in the numbers. We see that the underlying activities has increased, and but we also see that there was something extraordinary in the first quarter last year, receiving the milestone payment from Verrica, and also a bonus related to that milestone payment. We have a very strong balance sheet with approximately 180 million NOK in cash. And just to mention, in March, the PBM Capital notified the company that they wanted to exercise their warrants. But that share capital increase wasn't registered before April, so it's not visible in our balance sheet. Our quarterly report is now over per se, but we will continue as usual with the company presentation. And at the end of the company presentation, we will have a Q&A session. Over to you, Øystein.
Thank you, Gjest. So we are developing first-in-class molecules, which have shown very promising results in preclinical models and in cancer patients. And the way they work make them ideal for being combined with other types of therapies. We have recently completed one phase 2 study, and there are 2 ongoing studies in US with our lead candidate, LTX-315. The science behind our molecules have been validated by top-notch research institutions in Europe and in US, and acknowledged by the founder of the immune checkpoint inhibitors, who has become also our strategic advisor. Our technology is already commercially validated with a license agreement with the US-based company, Verrica, within skin cancer, and a US life science investor group with solid track record in biotech came on board as an anchor investor at our last capital raise. So as we have already touched upon, cancer is still one of the largest health problems globally, and clinical oncologists rank tumor heterogeneity as the major challenge in current cancer therapy. So how can we combat such a heterogeneous disease as solid cancer, which have a high number of different cancer cells with a unique set of mutations each? T cells may be the only solution because we in our body have a high number of different T cells, where each of them can attack one type of cancer cells. But in order for a T cell to be activated and respond, it has to be exposed to mutations, or more correctly, a tumor antigen. Oncolytic molecules have the potential to address tumor heterogeneity by their ability to generate a broad T cell response, which I now will show you. These figures illustrate how a large repertoire of different types of T cells become activated after LTX-315 treatment in an experimental melanoma model. The figure may be difficult to understand, but what is shown here is that a much higher number of different T cells are found in the tumor after treatment compared to the T cells in the tumor before treatment. Each dot is a unique T cell, and the brown dots represent different T cells types that are activated and increased in number. That an increased number of different T cells are found in tumors after treatment may explain why our drug LTX-315 is very effective against the same melanoma model shown to the left. A cancer model that is known as notoriously difficult to treat. And in the other melanoma model to the right, that is resembling melanoma in patients, neither chemotherapy or immunotherapy worked in this model. And it actually caught attention when almost all the animals in this model were cured by LTX-315 treatment. The two upper photos show the tumors that were not treated, and the bottom two photos show how the treatment with LTX-315 almost eradicated the tumors. And the survival curves show that the majority of the animals treated with LTX-315 survived, in contrast to the animals that were not treated. A following question is then whether the T cells can also be activated in cancer patients by LTX-315. These figures show T cell activation in a breast cancer patient from our completed phase 1/2 study. The figure is a bit different from the previous one. Here the x-axis represents T cells in the patient's blood before treatment, and the y-axis T cells in the blood after treatment. And each orange dot represents the unique T cells that is activated. And as you can see from the y-axis, a number of different T cells are activated in the blood after treatment. In fact, in this patient, 126 different types of T cells were activated and increased significantly in number after LTX-315 treatment. Activation of a number of T cells after LTX-315 were also found in other patients with different types of cancer following LTX-315 treatment. A critical question is then whether these T cells that are activated after local treatment with LTX-315 are able to attack other tumors distant from the treated tumor. In these melanoma patients, 2 tumors on the right side were treated with LTX-315, resulting in 82% size reduction of one tumor and 39% reduction of a second relative large tumor on the left side. And in this sarcoma patient, a tumor in the lower back of the patient was treated with LTX-315, resulting in a 63% size reduction of a non-treated tumor in the lung. And in the figure to the right, each column shows the effect on a non-treated tumors in one patient after treatment with LTX-315. And the figures show that in more than 1/3 of the patients, the size of non-treated lesions were significantly reduced with up to 80% size reduction as a result of local treatment with LTX-315. These results show that the T cells activated by local treatment of tumors are able to attack other tumors distant from the treated tumor in the body. There are several major challenges when it comes to cancer treatment, And one problem is that tumors contain different cancer cells with different set of mutations, and this is solved by oncolytic molecules by activating a high number of T cells that can attack many of the different cancer cells. Another problem is that the cancer cells can put on brakes or on the immune system that can block these T cells from working. Immune checkpoint inhibitors can solve this problem by holding off the brakes so the cells can complete the mission of killing cancer cells in the body. So by combining oncolytic molecules and immune checkpoint inhibitors, more patients could potentially be able to recover from their cancer disease. In a phase 1/2 study, LTX 315 was combined with immune checkpoint inhibitor pembrolizumab in a small arm with triple-negative breast cancer patients, which is a very aggressive type of breast cancer. In triple-negative breast cancer, the immune checkpoint inhibitor alone has shown some effect, however, in a previous study. But pembrolizumab alone did not show any effect on liver lesions in triple-negative cancer patients. Whereas LTX 315 in combination with pembrolizumab showed significant reduction of liver lesions in one of the patients tested. With their unique ability to generate T cells, both LTX 315 and LTX 401 have the potential to be an integral part of future combination therapies with immune checkpoint inhibitors in metastatic cancer. The global market for immune checkpoint inhibitors are expected to increase from $30 billion in 2020 to $140 billion in 2030. Still, the majority of cancer patients do not respond to immune checkpoint inhibitors, mainly due to active T cells. We have documented strong synergy with immune checkpoint inhibitors and LTX 315 and 401 in animal models, and promising effects with the combination in cancer patients so far. LTX 315 and LTX 401 may therefore have a large market potential in combination with immune checkpoint inhibitors. As mentioned, LTX 315 is currently being tested in 2 phase studies, and a 3rd phase 2 study is completed. And our 2nd generation molecule is in preclinical stage and will be made ready for clinical study in liver cancer patients within 2020. A 3rd oncolytic molecule has been out-licensed to a veterinary company in US for cancer in pets. When it comes to validation, commercial validation, we have already a commercial licensing deal in place with Verrica Pharmaceuticals, which in place potential future milestone payments of $110 million, in addition to royalty payments from low to mid-teen digit. Verrica has started a treatment in patients with basal cell carcinoma, and the collaboration with Verrica and their development program will be presented in more detail in a capital market day, June 1st. The promising results obtained with our oncolytic molecules is probably the reason why Jim Allison, who received the Nobel Prize in 2018 for the discovery of the first immune checkpoint inhibitor, chose to become a member of our scientific advisory board. And here he clearly described the need for drugs that can activate a broad T cell response to be able to attack all the different cancer cells in heterogeneous tumors, and the need for the right combination therapies to be able to fight cancer more effectively. The effect and mode of action of our oncolytic molecules have been scientifically validated by some of the leading cancer institutions in Europe and US. And we are very pleased to have the US-based investor group PBM Capital on board. PBM has a strong track record with their investment in successful biotech companies and is actively involved in Lytix and is also represented at our board. So to sum up, Lytix may have a solution to the most challenging problem in today's cancer treatment. We have already got much in place with regards to both scientific and commercial validation of our technology platform. We have a very competent board and a leadership team with experience from international biotech industry, both in Europe and in US. Our main focus is now to document that oncolytic molecules could solve this major problem in today's cancer treatment and be a part of more effective cancer strategies for the benefit of cancer patients. With this documentation in place, our clear goal is to bring Lytix to a new value inflection point. And with this, we have come to the end and are ready for questions.
Yes, we have received several questions regarding many of the things, activities going on. And I would like to start with a couple of questions regarding the Atlas IT-04 study. And first of all, the question is, are you actively working with Iovance to further your collaboration based on the finding in this study? And a similar question to that as well, have we shared the data with Iovance yet?
So to answer that, we have a non-exclusive clinical collaboration agreement with Iovance. So we will present the data for Iovance, but not before we have went through all the data, and that will probably happen after summer. And the second question was?
No, it's just regarding the data, if we have shared them with them, and if there is anything going on in this collaboration.
No. So we giving them regular update during the study, and we will now sum up the data and present to them after summer.
Good. Probably. And then we have a bundle of questions regarding Atlas IT-05. And I believe you might be the right one to answer those questions, Graham. And the basic question is, why did we make an amendment to this study? And what was the purpose of that?
Yeah, I'll be happy to answer that. One of our goals out of the Atlas IT-05 study is to document the effect of Lytix 315 in combination with pembrolizumab, and in a population where pembrolizumab has not been working on its own. So this amendment allows us to really increase the focus of the study in that direction. The melanoma population was already included in the study, but we had a broader range of tumors. So we're really increasing our focus on the trial. Another reason why we chose to make this amendment was to really give Lytix 315 a chance of showing an effect. But I will note that this is a clinical proof of concept study where we are trying to show that Lytix 315 increases the number of patients that benefit from anti-PD-1 treatment. But this does not mean that this will be the primary focus of the development moving forward, but it will give us clinical proof of concept. Thank you.
Thank you, Graham. There are two additional questions related to this. And the first one is if this amendment will lead to any delays in the study. Second question would be, have you also changed the dosing and the dosing schedule in the study? This one goes to you as well.
Yeah, no, I'll take the first question first. So it's still our aim to complete recruitment of the trial within 2022, which was within our plan. Whilst the amendment is a change in direction, we have changed our operational network, which is designed to further increase our ability to complete enrollment within 2022. In regard to the second question around dosing, So the dosing regimen was changed in Atlas IT05. It was changed in the original protocol, just to be clear, as well as in the amendment. And this really is that based on earlier learnings from our phase 1 and 2 studies and some of the translational research that we've conducted, what really has helped us understand how, when we administer Lytix 315, how it's having an impact on the body. In some early studies, there were some safety issues as we look to identify maximum tolerated dose. But we also wanted to understand how different dosing regimens and schedules really would impact the T cells and their activation status, and ultimately how that would translate to clinical responses. So we really feel like we've optimized the treatment regimen in this current study to really balance minimizing any safety issues that we may see, while certainly maximizing therapeutic efficacy. In previous studies, we did have several doses and dosing schedules, but we believe we're at the optimal one in this study, and this gives us great encouragement. And we did discuss this new dosing and dosing schedule with our scientific advisory board, who also agreed that they believe this is the optimal regimen to apply.
Thank you, Graham. We also have a couple of questions regarding the financial situation. The first one is more specifically on LTX-401 and the phase 1 study planned. And the question goes as this: how large and how costly will this study be? And is it included in the calculation of the financial runway? There's one additional question regarding the finances. Are you actively planning for the possibility to extend the financial runway? And it says here beyond third quarter 2023, and if needed, by putting on hold work on the LTX-401 and other projects. I don't know who should answer that.
I can at least start. The clinical phase 1 study with 401 is not fully funded with the current cash at hand. How expensive it will end up to be is not yet known, but I believe we have discussed 30 patients. Øystein will have to correct me if I'm wrong, in a monotherapy phase 1 study. Whether or when we start this would need additional financing, but we first need to prepare that molecule for the phase 1 study.
If I can add on that, we are working with designing a phase 1 study. It will be anyhow not a big study. It's a first safety study. So we will, during the next month, know more about the number of patients and the cost for this study.
The second question, I believe, was about run rate and whether we want to postpone 401. And as obvious from the answer from the last question, since 401 is not included in the current runway, we can't. Any postponement there wouldn't have a big effect. When and where, if we are going to raise more capital, that's up to the board to decide, and we will, of course, inform the market when that's happening.
Yes, and then a question regarding the announced Capital Market Day. That's for you, Øystein. Why are you focusing so strongly on the deal and collaboration with Verrica and the indication basal cell carcinoma at that event?
That's a good question. So we will have a Capital Market Day June 1st, together with the CEO and CMO from Verrica. And of course, the reason for our side is that basal cell carcinoma is a very large indication. And we are talking about 3 to 4 million diagnoses each year. The current standard treatment is surgery and could be quite problematic for the cancer patients. And we may have some new way of treating these patients that can be very a better treatment for the patients. So there we see a large commercial opportunity in this collaboration with Verrica, and the financial potential in the deal we have with Verrica is significant if they succeed, and that's the reason why we have a high priority for this collaboration with Verrica in our activities.
And this one goes to you as well, Øystein. It's regarding the amendment in the 05 study. Why was the study not, or the changes not publicly reported by the company before they were released on clinicaltrials.gov?
Yeah. So during clinical development, there is often need to do some changes in the protocol, so it can be several amendments. I think Graham has touched up into this about going from a more broader group of patients to a more homogeneous group. We have considered this as a not very high, not an impact on the value of the company, because the concept of this study is really not indication dependent per se, because it's really to show that 315 enhance the number of patients responding to checkpoint inhibitor. But with a more homogeneous group of patients, it can be more easy to value and analyze results. But we have discussed this, and we didn't find the need for making any press release of this, and especially because half of the patients that would be included in the previous protocol with anyhow be melanoma patients.
Thank you. And I have two more detailed questions there regarding the Atlas IT05 study. And one I think you would have to answer, Graham. It goes, could you explain a bit more on your cooperation with Thomas Madden at Mount Sinai and why this is valuable to the company? Did you get that question?
Yeah. I did, yeah. So Dr. Tom Moran is an investigator in the early phase unit at Mount Sinai Hospital. He has joined our study when we were looking for a broader population of patients, and was interested in participating as a result of our science. The study has moved focus to melanoma. He is working very closely with a physician in the melanoma department at Mount Sinai, Dr. Friedlander. So Tom Moran is really working in collaboration with Dr. Friedlander to bring patients into our trial.
And then finally, one even more specific question. In a recent article, a team of Chinese scientists proposed systemic administration of LTX-315 in combo with a CpG adjuvant and anti-PD-1 antibody to boost immunotherapy of melanoma. Is this something that you are actively considering going forward? Who should answer that?
So yeah, I can answer that. We see sometimes some groups out there doing some research on our drugs, and first of all, we have developed LTX-315 from a group of molecules in nature called host defense peptides. They always work locally at high concentration, and I think there are reasons for that because they have a high positive charge. So first of all, giving this systemically will create big challenges in the body, and they would probably have problem to reach the tumor. There could be certain ways of giving this systemically, but our strong belief is that, which we also will touch upon in the Capital Markets Day with Aurélien Marabel, talking about the rationale of intratumoral treatment. And that's because instead of giving a drug all over the body, risking toxicity and that the molecule don't in the end reach the tumor, you give it directly to the tumor and expose the tumor antigens. Everything is happening in the tumor, and you have to expose that for the immune system. So we, at the moment, don't really believe in a systemic administration of our type of drug.
Well, then, thank you to all of you for your answers. And I believe that we, by this, have ended the Q&A session.
Thank you.
Thank you.
Thank you all
Thank
for
you.
your attention.